Hey, Mr. Hillaby here. I'm going to cover the first part of the outcome you see here, explaining the formation and transmission of an action potential, including the all or none response and intensity response. And I'm going to focus on what an action potential is in this screencast. Okay, skip past that. All right, so a little experiment, FYI. Okay, they place a microelectrode into a squid axon, which are quite large, so that they could measure the voltage or the charge inside of an axon. And when they did this, Curtis and Cole determined that the inside of a resting neuron is negative 70 millivolts, which we call the resting membrane potential, or we say the membrane is polarized. So they realized that the inside had a negative charge and the outside, relatively speaking, had a positive charge. And when they stimulated the squid axon to send a nerve impulse through it, there was a quick reversal of charge that could be measured. And so that quick reversal of charge is known as depolarization, which we'll talk about later. We're going to be talking about it as an action potential or as a local nerve impulse. Okay, so what they realize is a nerve impulse or action potential is a change of the charge on the inside of the axon from negative 70 millivolts to a positive charge that lasted for a few milliseconds before it returned back to its negative 70 millivolts resting membrane potential. Okay, so the question is, what causes the resting membrane potential to be negative? So take a look at this image and see if you can come up with two reasons why the inside of the axon would be negative 70 millivolts. All right, so when you take a look at this diagram, there should be two features that you see that help you to understand why the inside of a resting axon is negative 70 millivolts. So this is the inside, negative 70 millivolts. Outside has a relative positive charge. So the two reasons why: one, you have these large negative proteins that are inside the axon but not on the outside, and those provide a relative negative charge to the inside. Secondly, you have more positively charged ions on the outside than you do on the inside of the axon. One reason is you have these potassium ion channels that are always open and allow potassium ions to diffuse from the inside towards the outside. And when they do that, they bring with them their positive charge, and that's going to, cause the outside to be relatively more positive, and it's going to leave the inside relatively more negative. So like I said, the membrane is on the inside is negative 70 millivolts, and we call that the resting potential or resting membrane potential. The other factor that's really important to understand is the fact that there is a higher concentration of potassium ions on the inside of the cell and a higher concentration of sodium ions on the outside of the cell. So as we start to look at what an action potential is, this is a key feature to remember. Okay, those ion concentrations of sodium and potassium when the membrane is at rest. And again, there are some potassium ion channels that are open that are allowing potassium ions to continually diffuse to the outside, contributing to that positive charge on the outside of the axon and leaving the inside relatively more negative 70 millivolts. As we work our way through what an action potential is, we are getting back to some Science 10 terminology, looking at, a cell membrane, and, phospholipid bilayer is selectively permeable. So it's got these gated ion channels that are going to allow and regulate the movement of potassium and sodium ions across the membrane in a process called facilitated diffusion. Okay, not only that, but we also have active transport. Like if you look down here, we have active transport of sodium to the outside and active transport of the potassium ions to the inside using what's called the sodium-potassium pump, which is a form of active transport, meaning it requires ATP energy by the cell. Okay, like I said, as potassium ions diffuse out of the cell, it brings a positive charge to the outside. Okay, again, large protein molecules that are negatively charged are present in the intracellular fluid, which is cytoplasm, but you don't find those in the extracellular fluid on the outside. So 2 reasons why we have a negative 70 millivolts on the inside of a resting membrane. Okay, and what this does is it creates a electrochemical gradient. Okay, it has stored potential energy in the ability to do work, which is going to be when you open up these ion channels, it's going to allow these sodium and potassium ions to diffuse or move. So how is this electrochemical gradient maintained? The electrochemical gradient is maintained by the sodium-potassium pump. So continually pumping sodium ions to the outside. Okay, and when you look at this image here, okay, we already have a higher concentration of sodium ions on the outside compared to the inside of the neuron. So this is why it requires ATP energy in a pump to pump those sodium ions against their concentration gradient from a low to a high concentration gradient. And likewise, okay, potassium ions, there's a higher concentration on the inside when the membrane's at rest, if you look at the picture. And again, to pump those potassium ions against their concentration gradient, that's going to take ATP energy by the cell to get this pump to pump those ions against their concentration gradient. Okay, so sodium-potassium pump maintains concentration gradient by moving in sodium and potassium against their concentration gradients by active transport. Again, a lot of Science 10 terms there. Okay, so excess positive accumulate on the outside of the nerve membrane and excess negatives accumulate on the inside. And so that's an electrochemical gradient. And we say that the membrane is charged and it's a polarized membrane. So that is a resting membrane potential. Now let's take a look at what a localized action potential looks like. So a nerve impulse is also referred to as an action potential. So when a neuron receives a stimulus, which is a type of energy, the cell membrane becomes more permeable to sodium ions than potassium ions. So what happens is when the stimulus arrives, sodium-gated channels open up, so they're closed when the membrane is at rest. So they open up, and this is like opening up the floodgates. You're going to allow sodium ions to start moving or diffusing through those gated channels, those proteins. The potassium-gated channels, those remain closed. So what ends up happening is sodium ions rush into the nerve cell. And so there are 2 ways we can explain why sodium ions are rushing into the nerve cell. So one is just explaining it in terms of diffusion, right? So ions will diffuse down their concentration gradient, which means from a high concentration to where there's a lower concentration. The other way to think about it is in terms of charge attraction. So if the inside has a negative 70 millivolt charge, so it's negatively charged, and of course sodium ions have a positive charge, then there is charge attraction. So those sodium ions will move to where there's a negative charge. Okay. In either case, we call this process depolarization. So one thing you need to be able to do is analyze a graph of the voltage of the inside of a membrane during an action potential. So when you look at the right-hand side here, we start at when the membrane is at rest. A stimulus arrives, which is right here on our graph. And then that opens up the sodium-gated channels. Sodium ions rush into this axon or into the neuron. And that is going to cause a reversal of charge, a brief reversal of charge, which figure A refers to this part of the diagram. So you have a quick increase and change in charge up to what we call maximum depolarization. And you could read that number on a graph if need be. So in this graph here, it's positive 40 millivolts. It's at maximum depolarization that you have the sodium-gated channels slamming shut. So that's going to stop any more movement of sodium ions across the membrane. And at the same time, the potassium-gated channels, they now open up. So the potassium gates open and potassium ions start to diffuse out of the nerve cells. So they're again moving down their concentration gradient from high concentration on the inside to a lower concentration on the inside. And that process is called repolarization. So again, figure B refers to B on the graph. So you can see what happens then is as time is going by and potassium ions are diffusing out of the axon, the inside then is becoming relatively more negative to the outside. And you can see we start to drop, okay, in terms of the negativity on the inside, becoming more negative. And you can see what's going to happen here is because the potassium-gated channels close slowly, this allows excess potassium ions to diffuse out of the axon. So they're taking with them their positive charge to the outside, and that leads to part C in our graph here, where you can see for a brief time that the inside of the axon actually drops below resting membrane potential, drops below negative 70 millivolts because the potassium gates are closing slowly and excess potassium ions are diffusing out. And we call this process hyperpolarization. So now essentially what we had is, if you think back to what just happened, okay, we had depolarization where sodium ions have rushed in. That's followed by repolarization where potassium ions have diffused out. This is my axon here, the inside. So essentially what's happened now is we actually have now high sodium on the inside and high potassium ion concentration on the outside. And that is the reverse of what we need during resting potential. And a neuron cannot fire another action potential until it has recovered and gone back to our original. Remember, what we're trying to get to is high sodium on the outside, high potassium on the inside during resting potential. So the sodium-potassium pump is going to continually, and it basically continually works throughout this whole action potential process here. It's going to continue to pump sodium out. Okay, pump sodium out and potassium ions back in. And that will reestablish resting membrane potential. And that time it takes to get from an action potential back to resting membrane potential is referred to as the refractory period. So energy supply from ATP maintains polarized membrane. Okay, the period of depolarization must be completed and the nerve must repolarize before a second action potential can be conducted. So the period of time required for a nerve cell to become depolarized is called the refractory period, lasting anywhere from 1 to 10 milliseconds. And if need be, you could read this off the graph. And I've seen it here where you measure from the beginning of depolarization. But on previous diplomas, I've seen them take it from the maximum depolarization. So read from there back to resting membrane potential. And this time period right here is the refractory period. So you could label that in your own notes. Okay, so that summarizes what an action potential is. So again, making sure we recognize the key terminology here and recognizing what is happening during an action potential. So number one, we have resting membrane potential. Okay, the stimulus arrives right here, this little blip here, right where we're starting to see a little bump. On a side note, okay, coming later in the notes is, and depending on the graph you look at, you might see a little bump here. So when the stimulus arrives, it will open up some of the sodium gated channels. So some sodium ion starts to rush into the axon, and there is what's called a minimal threshold potential, which is the minimal stimulus required to get a all or none response. So in other words, neurons have to have enough of a stimulus to open up all the sodium channels. And in this graph here, the minimal threshold potential, you can read it across here. So at this point right here is negative 50 millivolts. So if the stimulus is great enough that it opens up enough sodium ion channels to make the inside get to that minimal threshold potential of negative 50 millivolts, then at that moment, then all of the sodium gated channels open up and then you get depolarization. Okay. And essentially neurons either fire maximally or they don't fire at all. Okay, so we call this the all or none response. So as long as you meet that minimal threshold potential, all the sodium gated channels open up and then you get maximum depolarization here. Okay, and again, recall 3 is depolarization. This point right here is called maximum depolarization, in which case sodium gated channels slam shut, the potassium gated channels open up, potassium ions start diffusing out of the axon, making the inside relatively more negative. Because the potassium gated channels close slowly, then that means that you're going to get hyperpolarization of the inside. It's going to drop to below negative 70 millivolts or whatever our resting membrane potential is. And then the sodium potassium pump will reestablish high sodium on the outside, high potassium on the inside, getting back to resting membrane potential.